Uh, Good afternoon, everyone. As of in the morning started, everyone just listening the clinician. As a background, I'm not a clinician. I'm a stem cell biologist, and the core specialization mine in the bone marrow transplantation. I have started my career to isolation of CD34 40 positive hematopoietic stem cells and their delivery to the bone marrow. Because as we all knows that the delivery of the stem cell towards bone marrow is quite difficult for the engraftment of the cells. Then again, I have just changed my line, come to development of the cell therapy based the product. I have joined that Medi around 12 years ago. And at that time, my basic role to establish the facility, establish the R&D department and develop the product. And I have started my career as a cell therapy scientist. So then my basic role to develop the product, to regulate the product for a regulatory clearance from the so many notified bodies, governments, including the US FDA, Indian FDA, and the European bodies. So then we just brief about the Datt Medi. Datt Medi is a basically around 27-year-old company and basically dealing with the advanced wound care and a medical textile and bandages based product. Earlier, Datt Medi is a basically second largest manufacturing company around the globe for medical textile and bandages and dealing only in the European continent, not in India, not any Middle East part. But in 2000 now they have started that. And 2012 we have started the R&D unit developing so many biopolymer based products like nasal dressings, hemostatic dressings, silver based dressings, everything. But in 2012, when the company planned to start the cell therapy based product development, we have started mesenchymal stem cell based product development. And meanwhile, we have started some antimicrobial dressing because in the current world, mostly around the European part is basically stopped the using silver based dressing. Now in 20th December 2023, Germany has completely banned the silver based dressing in their part. So we have developed first the things. We have a lots of patents. Almost 22 US patent has been granted from 2012 to 2019 from the MISET, and two patent, that is the skin based patent and herbal patent, has granted as an excellent patent that grant within 3 months without any questions on that. So I directly come to the wound healing, as everyone knows about that from the till morning to the evening, that how the wound has been started and how it has been healed. So whenever the wound has created, first the hemostasis pathway has been started by regulation of so many cytokines, growth factors, and the clotting factor that promotes the hemostasis. Then it goes for the inflammation part, then cellular proliferation. From the cellular proliferation part, our job has come in the cell therapy section. So how we will proliferate the cells to faster healing sections. So what kind of growth factors or what kind of natural factors that can be provided to heal the wounds, and what is the basic role in the each kind of wounds that can proliferate in the wound healing. So that's where our job has come in that section. So then again, as per the CME part is the diabetic foot, as the diabetic foot has been come, and I think all of us knows about that. The foot wound has been developed by a dermis or the least part of the dermis in a person with diabetes. The underlying etiology of DF is classified as the three parts. Is the purely neuropathic. It's approximately 35% of the patient has goes for the neuropathic diabetic wounds. Ischemic, it's a 15, and again, it's the mixed population. It's a neuroischemic. It's a 50% population of the patient has come in that group. So as per the record in the morning, I have listened in the lecture. I've just not remember the name. It's the global diabetes. India has the global of the diabetic wound and diabetic patients in 2030. I think director has told in the morning. So as a thing, whenever the diabetes has initiated and has been increased, the wound always comes in the picture and the healing. The part of the things when the wound has occurred, we have used so many techniques to heal the wounds. Clinician has their own views. Clinician has their own style to heal the wound. Everyone has their own practice methods. But the treatment, what we feel that, I think Kari sir has just told about that the food, I think shoes that we are using that part. But how the healing has been started, we just forget how the wound healing has been started, how we will proliferate this process. This is the basic parameter that we want to increase on that line. So the pathogenesis of the wound healing, I don't want to add too much on that. It's too much ongoing. So we have using techniques. We are using a debridement method, their own debridement method, how to apply the products, how to use our products in the wound healing, and how it will affect maximum blood supply, how it will enhance the growth factors secretion on that, how it will start the proliferation of fibroblasts or keratinocytes or in the muscular cells in that area to heal the wounds. So that we have developed in such a way to include all this process in single product to heal the wounds. But as a scientist, I never find that any single product that will help us to remove the infection from the sites, or a single product that can use to proliferate the cells, that can enhance the migration of the cells towards the wound and increase the blood supply in that area. So we feel that it's a combination based therapy. We can use a combination of product to heal the wounds. First, we just control the infection from the site. After control of infection, we need to increase the blood supply. We increase the cellular migration on that, and then the wound has been started healing. So there is a lots of dressing in the market. As a company, definitely I want to say that my product is best, but it's not like that. There is a polymer-based product, there is a silver-based product, there is a gel-based products in the market that control the infection. There is a mechanical stability, a low skin adherence, and the pain elevation. It's a lots of product, but mostly that currently in the market, the product that present is the silver-based dressings that are used for the wound healing purposes. So that all these products that control the infection, all the silver-based dressing or existing product that present in the market just reduce the infection, but not proliferate the cells, not apply the growth factors or cellular migration on that area to heal the faster wound healing. So there is a dressing classification. There is a traditional dressings, basic dressings for that. That is the bandage-based dressing, cross-linking-based, the polymer-based dressing, like alginate-based dressing are used. Some chitosan-based dressings are also in the market. Collagen-based dressing. Collagen is definitely the best product that currently in the market to heal the wound. But the basic drawback with the collagen, that the adhesion property of the collagen, because collagen itself is protein, and the amino group of the protein will adhere to the pus, the material, the tissue that we secreted. When they apply the collagen-based dressing, when we go for a second-based dressing, it will again comes with the tissue that adhere to that area and cause a secondary wound and that. So that is the basic problem that we need to also remove from that area. So that is the bacteriostatic dressing, like a bacteria grass, silver-based dressing, honey-based dressing is also in the market, because as of I know, that is the mawa honey. That is the basically most of the European part has been used as a antimicrobial dressing that helps in the wound healing, but it can just only have an antimicrobial property, and it can provide a moist environment to heal the wounds, but it doesn't go for any cellular proliferation on that. So then comes the polymer-based dressing. Again, it's a amniotic membrane-based dressing is also there, but mostly amniotic membrane has been used as a graft, and with combination amniotic membrane and antibacterial dressing like silver-based dressings has been used to treat the wounds. Then we are in the picture for the velvet. That is a combination of two different kind of things. One, we are using a biopolymer. That is a chitosan and gelatin based biopolymer. We have made a matrix for that, and the matrix is an absorbable property that absorbs the exudates. It's approximately 20% of its weight. It can absorb the exudates. And as of chitosan, it's also a biopolymer, and it has a cationic in nature that binds with the gelatin and promotes the cellular proliferation. The cellular proliferation of the chitosan is well known, and it's already marketed. And then we have used natural products like curcumin, Amlica officinalis, and that the green tea extract to use as a cellular migration and treat the infection. So we have make a combination of these things and make a product. We have done the clinical trial in the burn wound, diabetic foot ulcer, and infected wound, and we found that almost 80% of the wounds has been healed within the 4 weeks after the application in the most severe cases. So after providing the support, after providing the dressings, we can find that the wound has been healed. So the time that currently used like a 50 days or 60 days in the like Acticode is a silver-based dressing that claim the completely wound healing of a diabetic wound is approximately 60 to 180 days, that we can reduce around 28 days. That has more social impact. The patient life has been good for that, and the cost effective again. So there is a so many dressing, and the bioactive dressing is like an Appligraph, that is a cellular fibroblast and keratinocyte based bilayered engineered product, the first cellular products in the come in the market. And the Appligraph has not a basically manufactured of this product. Organogenesis has developed this product in 1989, and the first commercialized cell-based product in 1999. After 10 years of the development of the product. And Appligraph is a definitely very good product. It used as a basically graft product that you can use for the healing of the wounds and a diabetic wounds and the venous leg ulcers. But it's a time around, it's a maximum time it will take. And the shelf life of the product is again, it's a compromise. It's approximately maximum 15 days. Then another product that's the cellular product is a dermagraft, that is the fibroblast based product that used for the wound healing, and is a very costly. It's approximately 21 square centimeter of the product comes around 1.5 lakhs. So that is the cost-based product. In that, that is the Kera skin. It's basically amniotic membrane-based product, and the new seal, that is again a product by organogenesis, and the healing time of that is again 120 days. Then we have developed the Wellgraft, is the same polymeric component, chitosan and gelatin, with the different ratio, and the porosity of the product has increased. Then we have used the mesenchymal stem cells culture on that with the adipocytes. So why mostly people are asking to me why you are using the adipocytes when there is a deep wound is there. When there is a deep wound is there, the healing time has been increased. But when you use the adipocytes with the any cells, it has been just reduced the time. And the best thing for this product to the scar-free healing. So whenever you use a adipocyte, it just reduce the scar and give a much better result in comparison to that. So then why we are using the porous matrix on that? When you are using a mesenchymal stem cell during a culture, mesenchymal will secrete a lot of growth factor, IL-2, IL-6, IL-12, and interferon. That will basically hold by the porous matrix structure. That will help in the cellular migration when you have applied, and that can basically faster migration of the cellular cells and faster healing. And in the Wellgraft, currently we are in the clinical stage, and we are doing the clinical trial in the SGPHA Lucknow, AIIMS Bhubaneswar, and the PGI Chandigarh, and everywhere where we have used the Wellgraft, we have get the complete wound healing. That is the chronic wound that will not heal last 2 years, 2.5 years, 3 years, that will heal within 28 days. So that's the clinical studies ongoing on that. I'm just showing some results on that. So I directly come to the results part. So that is the part that we have done it here in comparison to silver-based standard dressings with that, and that the result that we have found from the Wellbird. So and all the time point is at day 28. Maximum application time is the 4 application, and the observation time is at 28 days on that. So these are the trial data. That is the percentage of the wound healing that we have found. There's more than 69% patient has the 100% wound healed in comparison to the standard dressings. And we found that also the reduction of wound area and the time frame. So we are just evaluated how much time and how it will take to reduce the size of the wounds at the every visit. So this is the burn wound trial that we have done the PJMR Kolkata. So that again we found that in the burn wound, and in case of the burn, it's a personal experience, and with the clinician experience in the Safdarjung hospital, we are in the burn unit. We are mostly selling the Wellbird because after the application of the Wellbird, the sensation of the burn is reduced more than 70%. So mostly doctors has been basically recommended Wellbird for that after the burn. Then come to the directly Wellgraft on that. So it's a patented product. It's a complete US, India, and the European patented product, including the China also. And we currently tried for this product only in a diabetic foot ulcer. And that is made up with, as I already told you, the biodegradable polymer, titration and gelatin on that. And we have put it with the mesenchymal stem cells and the adipocytes. The seeding of the cells is a just a unique property. First, we have seeded the mesenchymal stem cell, then adipocyte. The time gap between the MSCs and adipocyte is also important. How to proliferate the cells and how they will fill the gap of the porous matrix. It's the important part of the healing of the wounds. And then we are using that. And the product that currently we release in the market, we have characterized for the all viral markers are the bacterial markers, and on the basis of a as per the complete international norm regulatory requirement to this. And the same trial has been already initiated in the US part for the Belgrade for the same diabetic foot ulcer indications. So these are the some patients data. The first one we have did it in the basically I think the 0 day 28 image has been changed. So it's AIIMS Bhubaneswar. Then the second patient's data, it's from the SGPJ Lucknow. Third one is again SGPJ Lucknow. And that the again fourth image, I think it's mistaken. And the fifth that we have done it here, that the complete wound closure within 21 days after the diabetic foot ulcer. I think that's the conclusion that we have found. And the basically velvet has been effective against all the superbugs and infection that we have tested. That will also help in the tissue cell migration and healing of the wounds, including the infection control. And Velgraf has been applied the completely non-infected wound, and it helps in the cellular migration and healing of the wounds.